DNA replication. How does the DNA copy machine work? Base pairing in the double helix explained how DNA could be copied or replicated because each base on one strand pairs with only one base on the opposite strand. Each strand in the double helix has all the information needed to reconstruct the other half of the mechanism of base pairing. Because each strand can be used to make the other strand, the strands are said to be complementary. Before a cell divides, it duplicates its DNA in a copying process called replication. This process ensures that each resulting cell has the same complete set of DNA molecules. DNA polymerase is an enzyme that joins individual nucleotides to produce a new strand of DNA. During replication, the DNA molecule separates into two strands and then produces two new complementary strands following the rules of base pairing. Each strand of the double helix of DNA serves as a template or model for the new strand. So the DNA polymerase is working and duplicating. It started here and moving this direction. And also here, the DNA polymerase works on this side and works the other direction. The two strands of the double helix separate and unzip, allowing two replication forks to form. As each new strand forms, new bases are added following the rules of base pairing. If the base on the old strand is adenine, then thymine is added to the newly formed strand. Likewise, guanine is always paired with cytosine. The result of replication is two DNA molecules identical to each other and to the original molecule. Each DNA molecule resulting from replication has one original strand and one new strand. DNA replication is carried out by a series of enzymes. They first unzip a molecule of DNA by breaking the hydrogen bonds between base pairs and unwinding the two strands of molecules. Each strand then serves as a template for the attachment of complementary bases. The principal enzyme involved in DNA replication is called DNA polymerase, which is an enzyme that joins individual nucleotides to produce a new strand of DNA. It also proofreads each new DNA strand, ensuring that each molecule is correct. That DNA polymerase also proofreads. The tips of chromosomes are also known as telomeres. The ends of the DNA molecules located at these telomeres are particularly difficult to copy. Over time, DNA may actually be lost from the telomeres each time a chromosome is replicated. An enzyme called telomerase compensates for this problem by adding short, repeated DNA sequences to telomerase, lengthening the chromosome slightly and making it less likely that important gene sequences will be lost from the telomerase during replication. How does DNA replication differ in prokaryotic cells and eukaryotic cells? Replication in most prokaryotic cells starts from a single point and proceeds in two directions until the entire chromosome is copied. The cells in most prokaryotes have a single circular DNA molecule in the cytoplasm containing nearly all the cell's genetic information. Eukaryotic cells, on the other hand, have up to 1,000 times more DNA. Nearly all the DNA of eukaryotic cells is found in the nucleus. In eukaryotic cells, replication may begin at dozens or even hundreds of places in the DNA molecule, proceeding in both directions until each chromosome is completely copied. In most prokaryotes, DNA replication does not start until regulatory proteins bind to a single starting point on the chromosome. This triggers the beginning of DNA replication. Often the two chromosomes produced by replication are attached to different points inside the cell membrane and are separated when the cell splits to form two new cells. Replication fork. The new DNA, the original replication started here and moving down. Replication fork again on this side, and here's the unreplicated DNA, and that is for the prokaryote DNA. Eukaryotic chromosomes are generally much bigger than those of prokaryotes. In eukaryotic cells, replication may begin at dozens or even hundreds of places in the DNA molecule, proceeding in both directions until each chromosome is completely copied. Here's the eukaryotic DNA cell, and replication forks in multiple places. Here's the unreplicated portions, and the DNA copying goes in both directions in multiple starting points. The two copies of DNA produced by replication in each chromosome remains closely associated until the cell enters prophase of mitosis. At that point, the chromosomes condense and the two chromatids in each chromosome become clearly visible. They separate from each other in anaphase of mitosis, producing two cells, each with a complete set of genes coded in DNA.